Good morning, respected sir. I wangtshen Tamang, bearing roll number 20NSO305, pursuing BSc Physics of 5th semester in Namsi Government College, affiliated under Sikkim University. The presentation is part of a sessional B, paper 7, which carries 25 marks. The topic of my presentation is types of Raman spectroscopy technique. Firstly, I'd like to thank our faculties of Physics Department, especially Dr. C.P. Khatiwara sir, for giving me such an wonderful opportunity to give a presentation on this topic. Introduction. C.V. Raman discovered Raman spectroscopy in 1928 to study the vibrational, rotational, and low-frequency modes of the molecules. It finds application mainly in chemistry to get the information related to fingerprints. The principle behind Raman spectroscopy is that the monochromatic radiation is passed through the sample such that the radiation may get reflected, absorbed, or scattered. Theory of Raman spectroscopy states that the changing in frequency during Raman scattering provides a basic for a form of spectroscopy. A general introduction is given to the principle and theory of Raman spectroscopy, the discipline dealing with spectral analysis of inelastic scattering of light by the vibrating molecules in liquids, solid, or gases. The phenomenon called Raman scattering, the origin of Raman scattering and its association with molecular polarizability is outlined in this article. The frequency relations of the vibrational Raman effects are developed in terms of the classical theory of electromagnetic radiation. A simplified description of the factor governing the directional properties, intensity, relations, and polarization characteristic of the scattered radiations are given by the classical theory, which are then refined according to a partial quantum mechanical treatment. The vibrational selection rules, including the role of molecular symmetry, are demonstrated using selective example of diatomic and polyatomic molecules. Contrasting infrared and Raman spectroscopy are emphasizing their complementary nature. Types of Raman spectroscopy: resonance Raman spectroscopy, surface enhanced Raman spectroscopy, micro Raman spectroscopy, and nonlinear Raman spectroscopy techniques. Resonance Raman spectroscopy. Resonance Raman spectroscopy is an advanced technique used to study vibrational bands in the group frequency region, and the information obtained is similar to that obtained by Fourier transform, infrared, and Raman studies. Surface enhanced Raman spectroscopy is a surface-sensitive technique that enhances Raman scattering by molecules absorbed on rough metal surface or by nanostructures such as plasmonic metonic silicon nanotubes. Raman microspectroscopy. Raman microspectroscopy is a powerful technique for analyzing both live cells and scaffold material used in tissue engineering. Nonlinear Raman spectroscopy is a spectroscopy based on nonlinear effects and involving Raman active molecular transition. Application of SERS, light source, NIR, VS, UV, subtracts various metal nanostructure, analytes single molecule detection, identification of a single DNA base molecule, protein, and cell. Principle. The surface enhanced Raman spectroscopy SERS is truly a surface selective effect. SERS consists in using the large local field enhancement that can exist a metallic surface under the right condition. Application of RRS, Resonance Raman Spectroscopy. RRS is well suited for the study of free radical transient species. It represents a great simplification which is focused directly on specific part of the molecules. Principle of micro-Raman spectroscopy. Based on the interaction between light and the matter in which the light is inelastically scattered. Application of micro-Raman spectroscopy used to measure the thermal conductivity of carbon fiber, carbon nanotubes, and graphene. Future scope. Imaging techniques in cancer, nanomedicine due to its high spatial resolution, high chemical specificity, and multiplexity modalities. Thank you.